All right, thank you everyone for attending today's seminar. I've asked Dr. Ondine Cleaver to present today. Ondine is a professor in the Department of Molecular Biology at UT Southwestern, and she's a great colleague and friend and mentor as well. So in terms of background, Ondine earned her PhD at UT Austin, where she worked under Paul Krieg on studying early blood vessel development, the role of VEGF-A and VEGFR2 on endothelial progenitors or endothelial cell patterning. She then did her postdoc at Harvard in Doug Melton's lab, where she studied angiocrine signaling and found that endothelial cells play an important role in the specification of, or in driving pancreas development. And then she joined UT Southwestern in 2004, and her lab has made significant contributions to understanding the mechanisms by which vessels form lumens. And I think this is really relevant to complex lymphatic anomalies as well as other vascular malformations where we see differences or abnormal lumens in the vessels. So with that, I'm going to turn the seminar over to Ondine and let her present her work.
All right. Thanks, Mike, for a nice introduction. And thank you all to be on the call today and to listen to me. Please feel free to interrupt at any time. I don't mind it at all. I can't see the chat super easily, so don't hesitate to just jump in if you have a question. So today I'm going to tell you about blood vessels and blood vessel formation during organ formation, and how critical the step of being able to make a central lumen to be able to carry blood for function of those tubes, function of the vasculature. I'm hoping this will be of interest to you all, since I know you're interested in maintenance of vessels and also in lymphatics, which are also tubes. So I'll jump right into things and just tell you that by background, I'm a developmental biologist, and this is really what interests me, is how do you make a tissue with form and function over the course of embryonic development, but really just any progenitor. How do you tell a progenitor what it's going to be and how it's going to associate with other cells to make an organ? That's really what has sort of driven me since I was a graduate student. And so I'm going to tell you about that today, both in vivo, and we're going to talk about some molecular pathways that are really important for making tubes, but then also what we can learn from that in vitro. And part of the reason for the in vitro work being interesting to me is that I've always been interested in how do you make organs, and more recently, I've been pulled into a number of efforts for trying to make tissues in vitro, make them in a dish. And part of the reason for that is that there's a great need for replacement organs. A lot of people get sick. Their organs fail, and they would like a heart transplant or a liver transplant or a lung transplant. And so the question is really, can we make those? And today we're going to really talk about kidneys and the potential for making kidneys. But the biggest question, and I think one that not a lot of people were thinking about for a while, was, okay, if we can make those tissues, what about plumbing them and attaching them to the rest of the body? And that would happen via the vasculature and also the lymphatic vasculature. But so our interest is that, can you make these tissues, and can you make the vasculature to fit with them? So the talk really will be organized into three main parts. The first part will be about blood vessel tubes. And what I'm showing you here is a very early mouse embryo, which would look almost exactly the same as a human embryo. And what you can't really see is the beginnings of the neural folds, which will eventually form the brain, the beginning of the heart. All of this green here, every single green cell is an endothelial cell. And these endothelial cells are the cells that line every single blood vessel. And what you're seeing in the bottom part of this little green blob here, the bottom part of this embryo, are these two very nice distinct vessels. And those are the first blood vessels that appear in the embryo, and the very first blood vessels that are actually going to open up a lumen to allow blood flow. And then in the second part of the talk, I'm going to talk about pancreatic tubules. And you may be asking yourself, what does that have anything to do with vasculature or lymphatics? But what I'm going to tell you is that we found that a lot of the molecular machinery that's critical for forming these early tubes is very similar between endothelial cells and the epithelial cells of the pancreatic tubules that also form tubes. And so these cells, even though they look different, they're very thin in endothelial cells, they're very thick in epithelial cells of the pancreas, but they know their inside from their outside. They know where liquid is going to flow, and the outside is where they're going to be attached to other tissues. And setting that kind of morphology up occurs very early, and it apparently depends on some similar molecules. So the third part of the talk, which is not indicated on this slide here, is going to be about the in vitro development of blood vessels and the challenges that we're going to face regarding being able to make any kind of vasculature in a dish. So let me start off with the blood vessel portion of the talk. And I just want to introduce you to our system. These are early embryos, and what I'm showing you here is the timing of their development, and we time it by the appearance of these muscle bundles along the back ridge of the embryo called somites. And these somites are going to end up forming major groups of muscles. And you don't really see them here. They're very transparent. But what you are seeing is all of these blood vessels that, and I meant to tell you this on the previous slide, these very dense blood vessels making it appear very green are actually the yolk sac of the embryo and not part of the actual embryo. It's continuous with the actual embryo, but it's not part of the embryo. The embryo is mostly dark at this point. But what you do see are these little speckled cells, and those are the progenitors that are going to form blood vessels. They're called angioblasts at this state, and they express our reporter, which makes it green, which is a GFP molecule driven behind the FLIC1 or VEGF receptor 2 promoter. And what you're seeing is really the emergence as the embryo has begun to gastrulate and begun to make mesoderm. These progenitors will appear and they'll start migrating around, like I liken them to little amoebas, but they quickly get corralled as if there was an amoeba cowboy corralling these into these little stripes. They find each other, they adhere to each other, they avoid certain regions. And we were really interested in why they avoid the regions such as the midline of the embryo here, or these little lateral regions. And that's all driven by actually concrete molecules that are expressed within the mesoderm telling them, don't come here. But what the ultimate result of that is that it ends up patterning them into these linear aggregates of cells. And you can see that here, they're really starting to take shape. And these two vessels are the very first vessels that are going to appear in the embryo, and they're called the dorsal aortae. There's two of them, and those dorsal aortae will slowly migrate and fuse together into a single aorta, which is what we all have, and the doctor tries to feel in your belly when he says, oh, I'm feeling your aorta back there. That's the largest blood vessel in an animal that carries blood away from the heart and to the periphery to all other organs. But so we're really interested in the formation of this early blood vessel. And something magical happens between somite stage 3 and the 5 somite stage, is that if you look carefully, you can see that this little aggregate here undergoes a change in morphogenesis. It goes from just a linear aggregate that's kind of like a rope, and it then becomes sort of like a tube. Oops, I touched my button without meaning to. But so this morphogenesis is called tubulogenesis, and it is the formation of a blood vessel tube. So let me just represent that again in 3 more different manners of looking at it. This is the view you had before. It's called a whole mount view. You're just kind of looking at the embryo. Here are all these progenitors scattered around. They aggregate together into these cords under the influence of cues in the microenvironment. And now they undergo this intrinsic change in conformation from a cord to a tube. And it's shown down here maybe a little bit better than my words in this cartoon, where you have progenitors, cord, and then tube. And here's a little bit more detail on that process, where it's a cross section through one of these embryos. And you can see these individual cells. They're very round. They're very migratory. They come together, and I false colored the junctions between these cells in red And I'm showing you what happens to these junctions as this lumen appears in the middle of this tube here, is that the junctions have to move away from this central canal to be able to open a continuous tube that's going to carry blood. And it's really a process I'd never really thought about until we found a molecule that was really a critical bottleneck requirement for this process. So the molecule that we found was actually the top of a long list of genes when we did transcriptional profiling of these isolated endothelial cells from a very early embryo. And what was interesting was in these long lists of genes that we got a hold of using at the time these chips, so microarray chips. And there was a lot of genes, and all of them had to do with small GTPase signaling. And small GTPases are cytoplasmic molecules that undergo changes from an active to an inactive state. And what they do is to catalyze molecular reactions within the cytoplasm. And this one gene that we found was called RAS-IP1, and it was highly conserved and it was novel. It was found really only in the blood vessels of frog, mouse, and zebrafish. We had mouse mutants that just lacked endothelial cells, the VEGF receptor 2 mutants, and they don't have a hint of RAS-IP1 expression, so it's very specific. And there was only one paper about it in the literature, and it was a paper by Natalia Mitten at UNC Chapel Hill. And she was a GTPase person and a RAS person, and she pulled out RAS-IP1 as interacting specifically with the active form of RAS, but not the inactive form of RAS. So this was exciting to us. Nothing known about this molecule, but it's very conserved. And so Kurzu, who was actually my technician at the time when I first opened the lab, it was just me and my technician in an empty room. And he basically took on the charge of helping me to understand what RAS-IP1 was doing. So what I want to remind you of, and this is going to be important during the talk, is that this molecule was shown by this very early single paper, not only to bind to RAS, but also to other GTPases such as RAP1, CDC42, and Rho, and that's going to become relevant in a few minutes. So let me just move on. So yeah, no known function. So Kurzu did a number of experiments. He knocked this gene out in vitro out of endothelial cells in a dish, and he showed that that impaired a lot of their normal behaviors, such as normally endothelial cells in vitro on Matrigel, for instance, will automatically just kind of get together and form cords, a lot like they do in the embryo. He also knocked out this gene actually in zebrafish and in frog. And both of those cases, it impaired the formation of these normal blood vessels, which are shown in purple here. So he had in vitro and in vivo experiments that showed that it was required. And then the next experiment he did was to knock it out of mice in my third year as a faculty member. And this was basically every junior faculty's dream, is an amazing phenotype that appears to target your tissue of interest. And so what you could see is in the heterozygote condition, you're looking at here a little embryo in its yolk sac, and there are large, beautiful blood vessels that course throughout both the embryo and the yolk sac, bringing blood to the embryo. And this is what we saw in our homozygous null, and the embryos die at this stage, E10.5. You can really see that this impairs the vasculature because the embryos essentially stop growing a day or two earlier, and they suffer from very severe edema and cardiovascular defects. And so a lot of my first 6 years as a faculty member was trying to work out, why these little guys were dying. And I'm really going to just spend the next couple of slides just summarizing all of that work. And the reason is that I want to move on to the pancreatic story, which is completely related to this vascular story. So I'm just going to give you the broad strokes of why RAS-IP1 is really important. So the phenotype boiled down to this, and I really previewed that in my introduction, but the ablation, genetic ablation of RAS-IP1 resulted in the failure of tubulogenesis. And so here I'm showing you a 3-somite stage and a 4-somite stage. And normally the evolution of these blood vessels, like I mentioned to you before, is they go from all these cells just all aggregated and adhered to one another to opening up this little lumen. And over time, I didn't show you this earlier, but these tubes just enlarge in size and scope. Their diameters get bigger. However, in the RAS-IP1, all of that just failed to happen. Some things happen normally, such as the cords form normally, the endothelial cells appeared normally, they were specified, they expressed endothelial markers. But what they were failing to do, that Kerr really found in his developmental cell paper in 2011, was that the endothelial cells failed to, they failed to change shape. So they remained very, very rounded. Unlike normal endothelial cells, I always say that the progenitors are kind of like an egg, which are very rounded, right? But if you take an egg and you crack it, and now you make an over easy egg out of it, the cytoplasm is like, the yolk, like the albumin, the white part, and it spreads out and it becomes very thin. And that's really what endothelial cells do. They become very thin. But when RAS-IP1 was there, endothelial cells had an inappropriate internal contractility, which kept them very rounded. So that was one thing they failed to do. And that had to do with their cytoskeleton. And I'll just describe that to you a little bit in a few slides. The other thing that they failed to do was to remain adhered to this outside stroma. Normally, they adhere via these integrins, and this completely failed. We showed this in vivo and in vitro. There's a hole that actually opens up regardless of whether or not you have a blood vessel. So the stroma, this stroma around these blue cells here, will actually become a real tissue. They'll become cartilage and bone. It's called the sclerotome. And that sclerotome has morphogenic power. And it turns out that it actually really actively pulls on these cells. So failure of this adhesion from endothelial cells to non-endothelial cells, failure of cell shape changes due to cytoskeletal and internal contractility. But when we published this paper, there was one question that we did not answer, which left the door open for new graduate students to come in and do some work. And this was the question we had not opened, we had not sort of touched on. And it was the failure in the RAS-IP1 nulls to clear these junctions from the surface that is eventually going to give rise to the lumen. So it failed to clear apical junctions is what it's called. So here, just to back up, normally, endothelial cells will have a basal side and an apical side shown in green. And what they do, oops, sorry about that. And what they do is that they will clear any junctions from this green surface. And here I'm showing you one of those junctions in red called Z01. And it clears it completely. And you can see a little hint of a lumen opening here. And the junctions go to the side. And the junctions are required because you don't want these blood vessels to bleed, right? Or to have any kind of hemorrhage. So keeping those intact is really critical. But in the RAS-IP1 mutant, you can see here that these junctions dot the apical surface, and they are also at the basal surface. To some extent, there was a polarity failure, but part of the polarity was also okay because the green surface was actually kind of between those endothelial cells. But so this apical junctional clearance became a major problem for us. And so a new student in the lab, David Berry, took on this problem to ask, how do these junctions get cleared out of the way? And that's a really, really important thing for these blood vessels lumens to actually be continuous. And so Kerr, who I failed to mention before, I go back, I just want to show him again. He was my first technician, also my first person in the lab, but he then joined the lab as a graduate student, and that his developmental cell paper was really his PhD work. But one of the experiments that he did, which was amazingly brave, and I'm so glad he did it, and I would not have done it as a classical developmental biologist, but he did basically a pull down to see what molecules were associated with RAS-IP1, because we just had no idea what RAS-IP1 was doing. And he pulled down two very important molecules. He pulled down RAS-IP1, but he pulled down RGAP29, which we knew nothing about, and another molecule called non-muscle myosin heavy chain 2A. And all these bands down here, you may ask what those are, and those are all going to be actin. So we're going to ignore those, even though we know that this molecule here, non-muscle myosin, is actually attached to actin when it's doing its job in the cell. So let me just tell you a little bit about this complex. RAS-IP1, from what we could tell, didn't really have any domains except for a RAS binding domain and a protein binding domain. It didn't really have other domains that we recognized. But when we realized that it was associated with these two molecules, we realized it was acting as a scaffold. So what are these two other molecules? So this big molecule up here, which is in two parts, you can see it has this long domain here and these heavy chains in purple and light chains in pink. And what this molecule is really the muscle of a cell. And when you activate this, it pulls together, it contracts, all right? And this is activated by phosphorylation of this pink domain, this myosin light chain. And when you phosphorylate this, it'll go ahead and contract. However, this phosphorylation event, I just want to point out, is dictated, is stimulated by the activity of a molecule called Rho. And it turns out that this other molecule, RGAP29, is a Rho gap, which turns off Rho. So I'm not showing you Rho here, but Rho will activate this top molecule and this molecule will inactivate Rho. So it seemed really interesting when we pulled these all out together. And so we started really testing lots of experiments I'm not going to show you here, but what is the activity of Rho? What is the activity of GTPases that bind to RAS-IP1? And what are their influence on non-muscle myosin heavy chain? And it turns out that a lot of the GTPases that RAS-IP1 binds impact this RAS-IP1, impacts a lot of these GTPases that in turn are regulating this non-muscle myosin heavy chain. So I'm going to talk about some of those pathways in just a minute, very quickly, but it's going to be really important to know that these molecules regulate this actomyosin contractility and therefore the cytoskeleton. So I just want to take you through one small sort of thread of these experiments. And this is the fact that RAS-IP1 is known to control Rho, which I've mentioned via RGAP29. RGAP29 was shown by a different paper to really its only function is to turn off Rho. So I'm building, as you can see, sort of darkened a little bit here, this arrow meaning it's suppressing. And Rho in turn activates ROCK, and this is all known in the literature, and ROCK in turn activates this phosphorylated myosin light chain, which activates this contractility. So in other words, this whole pathway is really important for contractility, but RAS-IP1 and RGAP are most likely, from the literature and from what we think, going to be turning this off. On the other hand, through some of our in vitro experiments that I'm not showing you, RAS-IP1 activates a different pathway called the CDC42 and PAC pathway, and these in the literature are all known to turn on phosphorylated myosin light chain and non-muscle myosin. So we had a big question mark. How can you have one molecule doing the same thing, doing different things, inactivating a pathway, but activating a pathway, but both acting on these adhesion clearances via the same molecule, non-muscle myosin. So just to show you really quick, one of the ways that we did this was to genetically knock out these pathway members, both on this side of the equation and on that side of the equation. And each case, what we're asking is whether or not these junctions get cleared from this lumen. And so here's an example of one. We knocked out CDC42, and we showed that it was really important for junctional clearance in these early blood vessels. We then went down the pathway to the next molecule on down, which is actually a kinase, PAC kinase, and we didn't have an allele to knock that out genetically, so we knocked it out using inhibitors and growing these little embryos in rolling bottles and culturing them along with this inhibitor, and then taking them before and after lumen formation and asking, is this member of this pathway really important for lumen formation? And we showed that just like CDC42, these junctions just don't move out of the way when you inhibit this. So these two experiments here show you that RAS-IP1 we know is somewhere upstream of CDC42, and this entire pathway appears to be really, really critical for movement of these junctions out of the way. All right. What I'm not going to show you is the experiments with Rho-A, but which did not impact movement of junctions and instead impacted lumen diameter. And what I'm going to do is to simply summarize all of these experiments with some cartoons and just some pathways here, and just reiterate everything I've told you and then tell you what molecules I want you to remember. So in the initial steps, which I mentioned about these angioblasts being very rounded, finding each other within the microenvironment of the early mouse embryo, they make adhesions with each other, they establish polarity, they start to build an apical membrane, and then they move these junctions out of the way. With all these genetic and pharmacological experiments, we showed that RAS-IP1 is upstream of this pathway, and that each one of these is really critical for activation of this non-muscle myosin and movement of these junctions out of the way. Now, the story I didn't really go over, just for time reasons, is that RAS-IP1 also acts in a completely opposite manner on non-muscle myosin. And I told you how that happens via RGAP29 and RhoA. So what is it doing there? Well, what we found is that at a later stage in the development of the blood vessels, you now have to reverse that. At first, you wanted non-muscle myosin to be active. Now you want it to be inactive. And why do you want it to be inactive? Well, as these lumens first start to open, it's actually exactly at the same time that the heart starts to pump. And when the heart starts to pump, this starts to put blood into these opening lumens, and that puts pressure on the apical membrane of these growing, expanding lumens. But these endothelial cells here, you don't want them to burst. And so they catalyze this formation of this actin cytoskeleton, which is catalyzed by this non-muscle actin. So how do you get the lumen to expand in size? You got to relax that. So RAS-IP1 now comes in and does a completely different job. It associates with different molecules. It acts on different pathways, even though it is still via non-muscle myosin, and now turns it off. All right. And now this results in lumen expansion. So lumen opening, lumen expansion, same molecule at the top and bottom, but two completely different activities. And we do that doing timed experiments in early versus late embryos. But so why did I go through the pain, this very complicated set of molecules? And why did I want to tell you this story? Part of the story comes from one of our faculty meetings. I was showing all of these over the years to the faculty in our department, and my chairman, Eric Olson, asked the question, this is really cool and everything for people who like blood vessels, but not everybody likes blood vessels. And, is there a RAS-IP1-like molecule that is important in other systems? And I told him with absolute assurance that no, that in fact there was not. And that, RAS-IP1 was really a loner, and it was, but then I left the faculty meeting and I realized, I was like, I better check to see if that's really true. That's what I think from the reading of the literature, but I'm not sure. And so I realized that RAS-IP1, its ancestor in Drosophila, is a molecule called cNu. And even though cNu diverged to give rise to RAS-IP1, there's a very similar molecule called Afadin, which I'll introduce to you. And Afadin, as it turns out, is expressed in epithelial cells. And that's where the story about the pancreatic plexus comes in. And so just to start this story, I want to show you what the pancreatic plexus looks like. And what you're looking at here is actually not the pancreas, because the pancreas tissue has been made invisible. What you're looking at are all the holes and all the tubes within the pancreas. And this is a molecule called mucin-1 that is essentially paints. It's as if you just ran some paint throughout all the tubes and it painted the inside of those lumens. You may be able to see, if your screen is good, that there's some really thick epithelial cells all around these, but it's been made invisible. So let me just show you that with cartoons to just take you through how does the pancreas develop. And this was all really discovered by another graduate student in the lab, Ali Villaseñor. So how does the pancreas develop? Well, it comes from an early mouse embryo in which the gut tube at this point is just a single layer epithelial tube from the mouth to the anus. There's nothing on it. There's no organs. It's just a tube. And then at about day 9 of development, all of the organs start to grow along the gut tube. So the lungs start to grow, the liver starts to grow, and the pancreas starts to grow. And so this is called the pancreatic placoid. And what this is a thickening of this epithelium and a stratification of that epithelium. So these cells start to proliferate and they start to pile up on top of each other. And it's very different than if some of you know about the lung. The lung is more like a real tree that grows out a big, first branch comes up, and then secondary branches, and then that goes from there and it elaborates from there. And the lung is really kind of a single epithelial layer, but the pancreas, on the other hand, appears as a bag of epithelium. And it doesn't like this state because all of these cells on the inside of the epithelium are not polarized. They don't have apical or basal, and they don't like that. And so they start to undergo these changes, including formation of microlumens. And these microlumens appear in these thick areas of stratification by doing what, to our eye, we realize is very similar to what the endothelial cells do. These cells start to change shape, and they start to rearrange their junctions. They clear the junctions from the apical portion of the cell where you're going to form a lumen. So in our eyes, there was identical systems, but there's some differences. So I'll show you. So eventually what happens to be able to shape the pancreatic gland, the tree, instead of just regular reiterative branching, what you have is that these microlumens will reach out and they will fuse with one another. And as this happens, the epithelium keeps growing, but it also starts to intercalate and to go from this very multi-layered arrangement to single layer arrangement. And I'm not showing you that off the screen. Here is when the pancreas is really huge, and it is back to single monolayered and also single lumened as a ramifying tree. So this is really how the pancreas develops. And our lab is actually just really independently interested in how this early progenitor organ results in the formation of things like islets. So let me just take you a little bit through the work in terms of how does a lumen form in the pancreas, and I'll point out the similarities with the vasculature and the differences with the vasculature. So here we're looking at a really close-up view of that stratified epithelium. And in the red marks E-cadherin, which just shows you every single cell. In blue are the nuclei. And in green, which looks yellow here because it overlaps with the red, in green is Z01, which is that same junction that I mentioned before between endothelial cell that needs to be cleared out of the way. So what happens when you form a lumen? There's a cartoon showing you that down here, but let me take you through it in real tissue up here. These junctions appear within the stratified epithelium a little bit after it forms. And what happens is that you have cells, for instance, that have these Z01 on multiple sides. Z01 is supposed to be an apical molecule, so it makes it very odd to see it studded in multiple different places. But as lumens start to form, these cells undergo cell shape changes, and you'll see one here cut in sort of a glancing section, which looks a little bit like a bottle cell. And bottle cells are well known in developmental biology as being cells that apically constrict. For instance, in the blastopore lip of the early frog embryo. So anyway, in 3 dimensions, these cells will do that coordinately, forming these bottle cells, accumulating their junctions at this little midpoint here where the lumen is going to form. And then it opens up these lumens. So you can see that in this cartoon here. Okay, so this is really interesting, and there are similarities. Just like endothelial cells, they're moving their junctions around. Just like endothelial cells, they're changing shape, albeit they're not becoming skinny. And so the differences are what the cell shape is, and the differences are that Z01, instead of sort of moving to the periphery in endothelial cells, is accumulating at the middle and then opening up a lumen subsequent to that. All right, so let's get back to RAS-IP1 and Afadin, and the reason why I wanted to tell you both of these stories in terms of understanding how a blood vessel forms. So what is the role of the RAS-IP1 cousin molecule Afadin during epithelial tube and lumen formation? So here they are, these 2 molecules, and I meant to go over this earlier, but RAS-IP1 really doesn't have many domains known except for a RAS association domain, a fork head domain, which is just protein-protein, but it's very generic. And then it has this little dilute domain, which we completely ignored in our first studies, but has to do with cargo transport. And here's Afadin, which also has RAS association domain, protein-protein domains, cargo binding domains, and then has a few extras, including binding to the cytoskeleton. This is a F-actin binding domain. So what we started doing is that Burfin Azizoglu in the lab just took a look in the stratified epithelium as where Afadin was, and she looked at these nascent lumens, and she saw that Afadin was right there on top of the nascent lumen. And in red, what you're looking at here is cargo that's being brought to this apical surface to try and open up that lumen, and it's inside the cells at this stage. And then as the lumens form, Afadin is right there poised, and mucin gets deposited on this apical surface. And so as it turns out, Denise Marciano is a faculty member on campus who happened to have studied the role of Afadin in the kidney and had the mice. And so we grabbed the mice from her and we made our own knockout mice in pancreas. And what I want to show you is that when Afadin is knocked out of the pancreatic epithelium, which you're looking at here, this is the beautiful, very complex plexus that I showed you before. Here's a cartoon of it showing you how it's really a three-dimensional net. And what you could see is that when Afadin was missing, you have all of these little balls. And we were like, okay, well, what does that mean? This is a whole mount view. Let's take a look in section. And when we take a look in section, here's a nice normal lumen, which is, you can see this mucin at the apical surface. These cells are apically constricted here. And this is what you get when Afadin is knocked out in sections. First of all, the cells just don't change shape. They start to accumulate this cargo that's meant to go to the apical surface inside the cell. So you can see E-cadherin lining a cell, and there's mucin. But it also fails to do the same thing that the blood vessels fail to do, which is to get junctions to the right place, move them around so that you can open up this lumen. So here you can see Z01 very nicely at this apical surface. And here's Z01 inside of one of these cells. And so, you know, the neat thing about being able to use the endothelial system and the epithelial system is that that endothelial cytoplasm is so skinny. We never saw these kinds of things in endothelial cells, but the epithelial cells allowed us a much fatter cell in which the cytoplasm is much more available to look at and to test things in. And so because there was cargo that was stuck in the cells, you know, we started thinking about the little domain, which I just mentioned to you, the dilute domain, which we completely ignored in our vascular studies. And so we started thinking about cargo and trafficking within the cytoplasm, and some molecules, some GTPases that are known to be involved in trafficking are the Rab molecules. And so what I'm showing you here is Rab11, Rab8, and both of them failed to properly localize subcellularly. And so normally, here's a nascent lumen. You can see that by the mucin 1. There's Rab11 dotted along. Well, it's everywhere when Afadin is missing. Here is Rab8, which is accumulated at these nascent lumens. Well, it's actually gone. It degrades when Afadin is not there. Here is CDC42, which is known to be really important for exocytosis and also polarity. And it's right on top of these nascent lumens. You see the yellow here. Here you only see green and red because now CDC42 is not exactly, or it's not anymore near the lumen that's forming. So subcellular localization is now being directed by Afadin. And so we thought this was really exciting because there was a lot of work done by this guy, Keith Mostov, who works in MDCK cells, which is a in vitro system. And he showed that all these molecules were really important for making a central lumen. So in his system, it replicates what we're seeing now in vitro. And what his system showed is that all these molecules coordinate together to bring a molecule full of apical membrane, full of apical cargo to where it's supposed to be. And what we can see now is that Afadin is upstream of all of that, which was really exciting to us. So just one quick little note that's separate, but kind of fun. I had a new graduate student that joined the lab, Haley Barlow, and she asked now, okay, GTPases are really critical for lumen formation. Well, are they? Well, she took a look at the pancreas with and without deletion of GTPases such as Rab11, and this is what it normally forms. This is a close-up here showing you this plexus all nice and attached to each other. And now this is what it looks like when you get rid of Rab11. And so this phenocopied in a lot of ways, both our RAS-IP1 mutant and our our Afadin mutant. So I'm just going to go through really quick the model, which I've just already mentioned. Afadin coordinates coordinates this junctional localization. It also brings that apical membrane all the way to where it should be to open up this lumen. It helps organize these junctions in the right place. And it's really important for molecules like CDC42. What I haven't told you about here is non-muscle myosin and this contractility machinery. And that's still an open question for us. On the flip side, I told you about RAS-IP1 and how critical RAS-IP1 was for bringing junctions and also for GTPase signaling, such as CDC42 and Rho. What I didn't tell you about was what about vesicles and RABs, and that's really the next step for us in terms of the vasculature. So I know, Mike, you told me to speak only for 30 minutes and you want lots of discussion. So I'm just going to zoom through about 5 or 6 more slides, and I really won't belabor it. But all of these mechanisms and molecules that I've told you about so far is because, I really want to understand intrinsically why, what signaling pathways are critical for making a blood vessel. But I also want to make blood vessels in these tissues. And one of the ways that people have decided that they can make tissues is by taking very naive cells, such as induced pluripotent stem cells or embryonic stem cells, put a whole bunch of signals onto it or media, and then they can turn them. We know how to do this. We know how to turn them into heart. We know how to turn them into pancreas. We know how to turn them into kidney. And so there's a beautiful Nature paper in 2014. And if you'll focus right here, it says a human organoid with a full set of renal cell types. And this was actually Melissa Little, who was a collaborator of ours. And we were in a consortium exactly that, to make a kidney in a dish. So, when we saw this and we saw that there were endothelial cells, a new graduate student joined my lab, Annie Ryan. And what I tasked her with was to characterize all the blood vessels in an organoid. And so she learned about the kidney, and she learned how to, culture them, and she learned how to take progenitors out of the kidneys or to use pluripotent stem cells, and how to make these organoids. And she made organoids, and she saw that they had blood vessels, and she was all set to go for her PhD thesis. And I said, okay, let's start characterizing them. And then she started coming to me with all kinds of technical problems. Well, they're very variable. I don't know what's going on. They don't look good. And she thought she was doing something wrong. But the more we looked at it, the more we realized that organoids in blood vessels are terrible. People report them as being present, but in fact, there's so much wrong with them. And, one day, Thierry in my office, she wanted to quit graduate school. And I told her, I said, listen, this is the coolest thing ever. Everybody in the world is making organoids. And it turns out the vasculature is terrible in them. This is very worthy of a report. And so she then took on the task, with her chin up now this time, to understand better the blood vessels, both in the normal kidney and also in the organoid. And what she found was that they didn't pattern properly. They didn't invade the glomerulus. They didn't express some of the markers that we are interested in. And more than that, they didn't have lumens, which was really kind of a critical part of the job of a blood vessel. And then with this variability, I convinced her to try and look at different stages of organoid development. And what she quickly realized is that blood vessels are born within the organoids very normally, just like they are in the embryo. But the organoids do not have a little heart, they do not have blood, and they do not pump into these blood vessels, and therefore the blood vessels regress. And so she showed very beautifully how there's a burst of organoid blood vessels that very quickly goes away. And, as a vascular biologist, this suddenly struck me, and I thought, this is not unexpected. We know this, that if you sort of occlude, blood vessel flow within the blood going into a blood vessel in the embryo, it will just regress. It will just die. And so she actually showed that. She showed that in these organoids and in early embryos, if you just choke them off from their blood flow, they just simply regress. And so since then, we have really found that the only way that we can really make viable blood vessels in organoids is to implant them so that they receive blood supply from a host. And so what you can see is when you implant an organoid, the host blood vessels actually invade and anastomose and give these blood vessels some flow, and it actually keeps them from regressing. And so we're making mice now to be able to study that interface between host and organoid blood vessels. But it doesn't answer everything, and I want to show you this. So here's these little organoids implanted under the kidney capsule, and they kind of spread out here. We know that when you do this, you get nice association of these host blood vessels, and this is just a closer up view of this. However, we started taking a look at, let's say, markers for arteriovenous differentiation, arteries and veins. And here's a very nice glomerulus that has been invaded now that it has flow by this capillary tuft. However, it has a venous marker both in the vessel that goes in and goes out. It's supposed to both be arteries. So we know that even though they have been given flow, there are still problems in their differentiation. So just to zoom through, what we're doing now is to build an atlas. We're doing single cell sequencing. We're doing spatial transcriptomics. We're just finding out all the different flavors of endothelial cells within a normal kidney at all different stages of development. And these are the people in the lab that are doing it both with their hands and with bioinformatics. And now we have amassed an atlas so that we can point to each one of these normal kidney vessels, the glomerular capillaries, the arcuate arteries, afferent and efferent arteries, the vasa recta, the cortical capillaries. And now we can ask the question, in these early blood vessels, are the right ones there? Do they pattern normally? Do they do their job? And this really is a 7-year effort that has culminated in a very nice atlas with very nice markers, and we're putting out that paper very soon. So I really apologize for going way over time. Here are my acknowledgements. Lots of funding by this Rebuilding the Kidney group, the Leduc Foundation. Leduc is a consortium I'm involved in, which has to do with maintenance of blood vessels and with vascular malformations, which I haven't talked to you about today because we're just at the beginning of this consortium. So I would love in the future, if anybody's interested in any of these molecules I've talked about today and how they might coordinate with molecules important in sensing blood flow and in maintenance of blood vessels. And I'll stop there, as I mentioned everybody directly during the talk. So thank you for your attention. I hope you're still there, and I hope feel free to ask me anything. I'd be happy to answer.
Thank you, Ondine. That was a great presentation, and I'll get the discussion started.
Okay.
And then everyone can just feel free to just unmute yourself and ask your question. But one thing that I had forgotten that you presented in the background was that RAS-IP1 interacted with GTP bound RAS more than GDP bound RAS.
Yep.
And we see mutations in vascular malformations, which lead to more GTP bound RAS. These are mutations in either NRAS, HRAS, or KRAS. Now, I was just curious, is RAS-IP1 functioning as a downstream effector of RAS, where you need the active RAS and then it interacts, or is it acting upstream?
I think it is a scaffold bringing this activated RAS where it needs to be. Okay. So, we've looked for domains. We've not found a GEF or a GAF domain on there at all. What we do know is that it binds to multiple molecules, and very similar to what I showed you with this ROGAP, this RGAP29, right? Bringing it in vicinity to the non-muscle myosin and regulating whether it's on or off, right? So in fact, I'm talking to a RAS guy now who's down in Florida. And it's funny because he says he's been looking at RAS, and he has done all the same experiments that we've done in vitro and in vivo, and he's done adult deletion of RAS, and he sees major defects in lumen formation and maintenance. And so we're really interested in whether or not RAS is going to be a major player in vessel maintenance and in malformations. That's the goal.
Right. Any other questions? Maybe I'll ask another one. I think that it's the other thing was the RGAP29. Have you gone through and deleted RGAP29, and does that result in vessel defects?
Right. So yes, we have. In fact, great question, Mike. we made a knockout mouse out of it using the COMP consortium, and we've knocked it out only though conditionally within the blood vessels. And the problem with that is really interesting because, you know, we work during a time frame that's very short. Vasculogenesis occurs really in the time frame of 3 to 6 hours. And those early blood vessels, as they're kind of first getting together in these linear cords and then opening up these lumens, are just turning on markers like VE cadherin and Tie2. So either with Tie2 or with VE cadherin Cree, we could only knock out RGAP29 after lumens have formed. And so doing that shows that it's really important in the sort of thinning of these endothelial cells and the expansion of lumens. And that's what sort of cemented our idea that this excessive Rho signaling was really bad for allowing cells to differentiate, endothelial cells to differentiate. So anyway, the short answer is yes, we've knocked it out. Yes, it's really important. And yes, it helped us once we had that phenotype to be able to understand and put together that big model of how all these molecules work together. Oh, yeah, I see a hand.
Hi, Ondine. This is
Hi.
Selan Catania from NHLBI. It was great
Hey.
talk.
Oh,
And I
thank
missed
you.
a part of your talk, so excuse me if I'm repeating
no.
this or it is. So I was wondering if these mice have hypertension, first of all.
It's a great question, and we have not tested because I'm a developmental person. But God, we should really do this with somebody with
yes, you should do
hypertension.
it. Yeah.
You know, I mean, you know, it's funny. You're not the first person to ask me, and we should really check this, because what's interesting is that if you knock out RAS-IP1 at like 8.5 or 9.5, I mean, it kills them, right? Well, if you knock it out at 7.5 or 8.5, no blood vessel lumens open. If you knock them out at 8.5 or 9.5, the vessels are very constricted.
Yeah.
13, 14, still, you know, it affects the blood vessels, but the later you go in development, the less effect it has. And if you knock them out at 18.5, there's no effect. If you knock it out in the adults, there's no effect, other than the fact that if you try and do a little angiogenesis assay, sort of like cancer, right? You put a little matrigel plug under the skin, the blood vessels don't go in as well. So they're clearly impaired in their ability to be activated and to grow. In terms of the hypertension, never have done it, but I really appreciate the question, and you reactivate something in my brain about.
Yeah, and you can write more grants, you know.
I know.
Yeah, and we didn't, if I can carry on just a little bit, did you
Please.
isolate the vessels and test their contractile responses with any kind of you know, either calcium or yeah, I think you have a
Never tested.
Oh, please do so.
Okay.
Yeah.
No, I love it. And, you know, in fact, you say this, and there's a guy here on campus, Phil Shaw, who has a group that's really, there's a whole hypertension group. So I have experts right here. So
Perfect.
that
Yeah.
perfect. Thank you so much.
NHLBI loves collaboration. So
yeah.
All right. Thank
Thank
you.
you. Appreciate it. All right.
Well, if there are no additional questions, thank you, Ondine, for this great presentation. And we look forward to seeing everyone at our next seminar next month.
All right. Thank you, everybody. Thanks, Mike, again. Bye.